Now this is our tissue culture lab. And tissue culture is when you take pieces of a plant under sterile conditions and do interesting things to it. Either shoot proliferation, make a lot more plants, is a common technique in the nursery industry, or you can do CRISPR-Cas9 gene editing, which is a targeted mutagenesis, changing the genes that the plant already has. Or you can do genetic modification, which means introducing genes that you want the plant to have, but which it doesn't. And there's three basic methods that we use here. One is the classical transform your leaf disc, and that's with Agrobacterium tumefaciens. Another one is the gene gun, where you shoot genes into your tissue. And in our project with chloroplast engineering, that's what we have to use. It's the only way to do that type of method. And then our very exciting one is our root transformation method, because this is very, very quick. We're able to go from a root transformation to a plant with seed in only about 150 days. So it's extremely quick. In many cases this would take a year, and most species won't let you do root transformation because the tops they put on are not transgenic or not edited. This is an exception. So the other thing, as I say, is root proliferation and shoot proliferation, making a lot of plants clonally. And as you saw in the greenhouse, we could do it with root clones, but we can actually make a lot more under these conditions. So I'm just going to go through a series so you can see what the sort of thing that we do. So first, we would have some sterile seed and we would grow sterile plants. So here's a nice set of sterile plants. There's shoots on them and there's roots on them. Now we take those roots off under sterile conditions and spit them out and put them into new media. You don't need hormones for this, and you see these pieces of root have already started to put shoots on, and those shoots will be genetic copies of their roots. Now at that point we would then grow those up, and you might end up with something like this, where we now have a lot of shoots. Now this is getting a bit crowded, so you make a decision at this point: do I want more of these, or do I want to put these in the greenhouse? But in this case I'm going to say we want more, we don't have enough. So this box would get split between multiple boxes for proliferation. So we take those, and those would get converted to many boxes containing something like this, where there's just a very few plants. Now these are going to grow up again and fill the box. And then we end up with this box. Again, lots of shoots. Now I'm going to say at this point, these are ready to go out to the greenhouse. But these have spent all their lives in 100% humidity, except when they were being subcultured. So if I just plant these out, they're going to die of transplant shock. So instead, we'll subculture these into peat pellets. And then this is a peat pellet, and you can see the plants are doing pretty well. And so this one is about ready to go to the greenhouse. And what you first do is crack the lid to lower the humidity. You're not doing anything else. And we can then, after a few days with the cracked lid, then it'll be ready to go into a greenhouse situation. And this one here, you can see this lid is cracked open, and this plant will be potted up in the next week or so. Now, as you saw in the greenhouse, if you have a lot of dead leaves, this one's got quite a few dead leaves, this is bad for the plant. And if it's in the tissue culture, it doesn't matter so much in the peat pot, it could end up killing the plant that you want. So what we're doing over here, Nikki is in the sterile laminar flow hood, which keeps all of the air in front of her sterile with no fungus or bacterial spores. She is cleaning up a tissue culture with dead leaves, which are basically becoming toxic to the culture, to the healthy leaves. And then once that is done, she will subculture the healthy plants into separate little plantlets in the new media, in the new sterile magenta box.